नमस्कार, हमेशा खुश रहो बिटिया. I am Jv'n Dr. Lily Trivedi, and today I am going to take the lecture of B.Sc AG second sem students, and the course is Fundamentals of Crop Physiology. And the lecture today is Fatty Acids Synthesis and Breakdown. Now, we have already studied in the previous lecture fat metabolism, and now we are going to further study fatty acids and their synthesis as well as breakdown. Now, fats are hydrolyzed into fatty acids and glycerol by lipase enzyme. Now, further the fatty acids are converted into acetyl coenzyme A by the process that is beta oxidation. The acetyl coenzyme A is further converted into sucrose via the glycoxylate cycle, and then it is transported. So as already said, the fatty acids are broken down by beta oxidation. Now it reaches the Krebs cycle. The acetyl coenzyme reaches Krebs cycle, and further degradation and release of energy is there. But if you have glycerol, so it directly enters the respiratory channel by glyceraldehyde. Now the synthesis of fatty acid, it occurs in cytoplasm and endoplasmic reticulum of the cell. It is chemically similar to beta oxidation process, but with a couple of key differences. Now what are these key differences? First of all, these occur in preparing the substrates for the reaction that grow the fatty acids. The transport of acetyl coenzyme from mitochondria occurs when it begins to build up. The two molecules can play a role in moving it to the cytoplasm citrate and acetyl carnitine. Joining of oxaloacetate with acetyl coenzyme A in the mitochondria creates citrate, which moves across the membrane. Now, this is followed by the action of citrate lyase in the cytoplasm of the cell and to release acetyl coenzyme and oxaloacetate. Now, what happens additionally when the free acetyl coenzyme A accumulates in the mitochondria? It may combine with carnitine and be transported out to the cytoplasm. Starting with the 2-acetyl coenzyme A, one is converted into malonyl coenzyme A by carboxylation and catalyzed into the enzyme, catalyzed by the enzyme acetyl coenzyme A carboxylase. It is the only regulatory enzyme of fatty acid synthesis. Next, both the molecules of coenzyme A portions replaced by a carrier protein which is known as acyl carrier protein, and it forms acyl carrier protein and malonyl acetyl carrier protein. So these two are formed. So joining of fatty acyl ACP, in this case, I'm using the short form ACP for acyl carrier protein. So in this case, the acetyl ACP and with malonyl ACP splits out the carboxyl which was added and creates the intermediate at the upper right. Now you can see this in the figure also in the upper right. Now here is the acetyl coenzyme A. So here we have the acetyl transferase and it is converted into coenzyme A. So, and this is the acetyl coenzyme phosphate. Now what happens here? You can see these are the reactions. It is, as already mentioned, now it is converted into malonyl coenzyme A and acetyl ACP. That is a protein complex. Now this is further NADPH. It combines an NAD with beta ketoacyl ACP. Again, it is converted into beta hydroxybutyryl ACP. So these are the further reactions which you can see over here. And now let's go into detail what happens here. The chemical reactions which resemble those of beta oxidation are reversed. So what is the process? First, ketone is reduced to a hydroxyl using NADPH. Now, in contrast to a hydroxylated intermediate of beta oxidation, the beta intermediate here is in the D configuration. Now, there are two configurations we all know, is the L and the D. So, this is the D configuration. And next, what happens? The water is removed from the carbons of 2 and 3 2 and the 3 of hydroxyl intermediate to produce a trans-bonded, double-bonded molecule. Now, at last, the double-bonded is hydrogenated to yield a saturated intermediate, and this process cycles in the addition of another malonyl ACP is added to the growing chain, which ultimately an intermediate with 16 carbon is produced, and that is palmitoyl coenzyme A. Now, at this process, the fatty acid synthesis in the cytoplasm synthesis ceases. So, students, I hope you are satisfied with my detailed session. If you have any query, please write in the comment box and I'll resolve it soon. This session was powered by digital version 2.0 of Jyoti Vidyapeeth Women's University. Thank you and namaste.